Now again, this is called a what? What are we trying to build? A cladogram. What's another way? Oh, another word for cladogram? Cladistics is a study of this stuff, but what's another word for this? Phylogenetic tree. So it's basically a family tree of the relationship, the evolutionary relationship between organisms. We're going to use everything that we know, all that evidence of evolution, all the idea of analogous and homologous structures, all DNA evidence. You'll see things like DNA evidence coming into play, structural differences and similarities, how they eat, what they eat. These are all things you can use to classify organisms, right? If something's a carnivore versus an herbivore, for instance, that says something about their evolutionary history. How much it says and what importance you give it all depends. It has to be contextualized. What is more important? It all depends. So they made one chart, and I think that's the chart you have to start with, is characteristics. And in this example, they use snowflakes. So we're going to quickly do this, and we're not going to spend a lot of time on the snowflake lab. I'm just going to outline it here, and then I'm going to expect that you're going to upload it, and I'm going to see whatever it is that you actually accomplished with this. Because I'd like to get onto the biology example, which is the last section in this cladistics unit. So we have table defining the character states, right? So we have character, we have a character. What's another name for a character? Trait. Trait, or what's something else they might use? Phenotype. Okay, so somebody might use phenotype, character, trait, right? Yes? Okay, so we have a character, we have a description of the character. And then we have a state. Either it's 0, it's 1, or 2. So they give you a possibility of having 3 states. So it's not just on or off, maybe it's in between, right? So there you have it. That's what they gave you. You don't have to use any of this. You don't have to use all of them, but you definitely need to have at least 2, right? Yes or no? It's always preferable to have just 1 or 2. But obviously they're giving you a hint that you're going to need more than 2 in some cases. So character 1, 2, 3, let's just do 4 of them. There's 27, I think, right? 27 or 26, what was it? In the activity, did they have you do 27 or 26 of these? 26 of them, all right. So, knowing that that's what you have, what's the first character someone's come up with? You said big versus small. Is that right? I got a problem with that. What's the problem with big versus small? Is it big or not? Where do we draw the line on big? It's yes or no, right? One or the other, on or off. How do you choose then? If you say big or small, what problem do I have using big or small? Yeah.
There's not like an exact measurement for big or small.
Not enough. It's not enough to say big or small. Like yesterday, we were talking about a dime being small. Remember that? And we said smaller than a dime or bigger than a dime. That's easy. We can say, is it bigger than a dime? Yes, then it is. Is it smaller than a dime? No. Or smaller to or equal to the size of a dime? No, then it's no, right? So it's yes or no. When you say big or small, it leaves too much ambiguity. It's just like when we did color yesterday. It was either silver or it wasn't. You can't really do like, it's silverish, but then this one's kind of in between copper and silver. it gets difficult to make a cladogram if you start to have these non-binomial choices, right? Non-binary choices. Does that make sense? So it's best to go with either on or off. In fact, so much so that I'll tell you, just erase this, even though it's useful for this exercise, let's just get rid of this. Just to emphasize to you that what you really want to know is, does it have that trait or it doesn't have that trait? All right. All right, so give me a trait, guys. Good guess, though. The big or small, I would have said the same thing. You know, if I were you, if I were in your shoes, I might have said big or small, because I can see that there's a lot of big ones and then there's a lot of small ones. But then again, there's a lot of in-betweens as well. Yeah.
6 pieces coming out of it.
Six branches.
Yeah.
6 branches. So a 6 branch. So can we call them root branches? Huh?
I'd have called them
Arms.
arms.
Okay. So, did you distinguish between the arms and like the stuff in between the arms?
Yeah.
Okay. So you said branches or arms. Okay. All right. So, are there any other choices? Like I see 6. Is there anything else other than 6?
7.
So they have a 7. So they 7 branches. There's 6, 7, and there's one with 8 branches.
Sorry, I miscounted. 8 has. Sorry, 8 doesn't have 7 branches. 8 has. I meant O has 8 branches. I'm sorry, I counted that wrong.
Wait, you.
It's 8, not 7. Sorry, I messed up. It does not have 7 branches. It has 8. I accidentally miscounted.
So there is no 7. There's just 8. Is that what you're saying?
I accidentally miscounted. Sorry.
Oh, it's not a problem. Let's move on.
Q has like 12.
Yeah, Q has.
So we can go with 12 or 12 branches. All right. Anything else? How about something connecting the center? Can we say I don't know. Anybody have a name for it?
Diamond center.
Diamond center. Okay, it's not. They're not all diamonds. There's some that are stars. Connective. Can we call it connected center? Is that fair to call it a connected center? All right, so connected center. What else are we going to call it? What else? What's the next one? 5.
Non-visible center.
Not. You mean no center?
Yeah.
So it's either connected or it's not connected. So we're good with that, right? If it's not there, then there is none. Yeah.
If it has circles on the end.
Circles in the end. End caps.
Yeah.
Anything else? Yeah.
If they have lines in them, because O doesn't have lines.
What do you mean by lines?
So like, basically O is the only one that doesn't have like any lines coming from it. It's just like a whole lot of shape, but everything else is pretty like.
So shape. Everything else has like an arm, and O doesn't really have an arm. It's different, and it's a bunch of squares. Yeah, right. Can we say no arms? What do you think? Will that satisfy that characteristic? No arms.
Diamonds are long.
So you think it? There. Which one are you thinking about there? F. I see. So a bunch of shapes. In this case, diamonds up and down the arm.
Oh, definitely. They both have diamonds.
So diamonds. Diamond arms. Is that fine? Either has it or not. So you can continue this, and obviously they give you 26 of these because you can continue them. And then once you have them, then you can decide where you want to put your tree. So of course, if you're going to have your 0, 1 states, and you have you're going to need a chart that's going to allow your different characters, your different taxa, A, B, C, D, right? A, B, C, D, E, F. Let's just do A, B, and C, A through F really quickly. A, does A have, let's say, character trait? Let's go with character 1, and 2, and 3, and 4, and 5, and 6, and 7. Of course, you can do all of them, right? But I'm just doing the first 7 here. So 1 through 7, does A have 1? Yes or no? On or off? 0 or 1? So 1, everybody, does everybody agree that A has character 1? So that's 1. Does A have character 2?
No. So 0.
Does A have character 3? Does A have character 4? 5? Character 5? Does A have character 5?
No.
Does A have character 6? Yes or no?
No.
7? B, does B have character 1? Yes, it does. Does B have character 2? 3? 4? 5?
No.
6? 7?
No.
Does C have character 1? I can't hear you. 2?
No.
3?
No.
4?
No. Well, no.
There's only one person doing the lab. 6 or 5? 6? 7? D, does it have character 1? 2?
No.
3? 4? 5? 6? 7? E, does it have character 1? 2? 3? 4? 5? 6? 7? F, does it have character 1? 2? 3? 4? 5? 6? 7? All right, so if we're going to place these anywhere on this chart, now what problem do we have? Our problem is that because we don't have enough character states, right, characters, because we don't have enough characters, we have too many that are too similar. So you got this one is indistinguishable by this chart, quantitatively indistinguishable from each other. Do you agree? They have exactly the same pattern. So there's no evidence for me to separate this taxa. The fact of the matter is, if I was forced to draw something based on this, I would say these are the most similar. Do you agree? So I can take and put a branch on here. Everybody has 1, so this is trait 1. Then I can put a branch out here that has trait 1, but they also have trait 4. And from that branch, I can get, assuming that they're actually different, two branches. And then this one can continue on and separate here. So there's trait 4. These two, B and E, have trait 4, but the interesting is that they're not like F, are they? Because even though they have trait 4, they also have, F also has trait 5 and trait 7. And so that would be F. Do you see that? So B and E have trait 1 and 4. B and E have trait 1 and 4. Do you all see that? But so does F, but F has 5 and 7 as well. So now we're left with B, I'm sorry, we got, which ones did we just do? We did B, E, F, A, C, and D. There's no real way for us to distinguish between them because simply because we don't have enough characters to distinguish them. So we could do A, C, E. A, C, D, sorry. You see? Based on this evidence that I have here, limited evidence, this is the cladogram, the relationship between these snowflakes. That's the best I can do with the data I have. If I had 26 different characters, though, I could probably separate them a little better. I have one that they all share. Everyone has character trait 1, but this branch has developed characters trait 4. That's these two. These have 1 and 4. And F has 1, 4, 5, and 7. Are we good with this idea of how to read these cladograms and how to create them? So I expect by tomorrow that you guys have a cladogram on snowflakes that takes into account as many characters as you can think of. Is that fair? All right. So let's move on. Now I want to say something before we go on, and that's that I can circle everything with a common ancestor. I can circle this group right here. Do you see how I can circle that group? That group, I can call this a clade. I can call it a clade. That's why we call these cladograms. Because I can say all of the creatures, all the snowflakes in this case, that share characteristic 4, I'm going to call it clade. It's much better than choosing to call it a mammals, let's say. Why would it be better than calling them mammals?
They're not mammals.
Okay, let's assume these were. Let's assume these aren't snowflakes, that these have hair, all of them are warm, are homeothermic, all of them have mammary glands and provide milk for their young. Why not just call them mammals? Why is it better to call it a clade and just look at the characteristics that we're looking at only?
Giraffes give milk to their babies.
Yes, they do.
They have nipples.
Yes, they do.
I mean, I know there is it.
So do whales. I think whales are the more interesting beast that has breasts, don't you think? Yeah. Do you all agree that here's what I'm going to ask you? If we just defined, if these were mammals, they're not, they're snowflakes, right? But if they were mammals, the reason that it's better to go ahead and use this cladogram and call them a clade instead of calling them mammals is you run into problems. Can I give you some examples? So we call mammals things that give live birth, like humans, right? Okay. Well, what about things like the duck-billed platypus that lays eggs?
It's an exception.
It's still, well, but it's a problem, isn't it? You can't say all mammals give live birth since oviviparous mammals, like the duck-billed platypus, lay eggs. What about things that, all mammals, what's another example? All mammals have placentas, right? So we have placentas. Shouldn't all mammals have placentas? Well, they don't, right? We know we have what type of mammals? Kangaroos, and what are they? Marsupials. So marsupials are a problem to that system. Let's say all birds fly, right? Oops.
The ostrich don't.
Yeah.
Penguin.
Penguins don't. Ostriches don't fly. It does. Turkeys do fly. But penguins don't. Penguins swim. So you start to run into problems when you start to say all this or all that. So the best way to do it is to look at the characters you have and group them based on the characters that you have. Don't depend on what you know, because what you know might be wrong. Look at the characters, tabulate them, and make decisions based on the evidence that's in front of you. That's why cladograms are how we classify organisms. We still use the Latin binomial system. We still use Homo sapien, for instance, or Rattus rattus, or canine, right? But we can create a clade where these organisms share character traits. We just look at the character traits. Just be as plain and simple as you can. Occam's razor, right? Keep everything as simple as you can and compare the characteristics and make decisions on groups, not based on some definition that we find in a dictionary, but based on the evidence that you have right in front of you. Does that make sense? All right. Phylogeny and cladistics activity. It talks about glyceraldehyde 3-phosphate dehydrogenase, which is an enzyme, right, that catalyzes the 6th step in glycolysis. Do you remember what glycolysis is? Breakdown of sugar. Very good. Into pyruvate. Excellent. The following data shows the percentage of similarity between of this gene and expresses in humans versus other species. So this is gene. I think we'd all agree that roundworms, fruit flies, dogs, and chimpanzees are very different. We'd also, since they all do glycolysis, we expect that they're going to have the enzyme, glyceraldehyde 3-phosphate dehydrogenase. Do we all agree that you would expect to see the enzyme? But knowing that, remembering that DNA mutates, remembering that mutation leads to changes in amino acids, that those changes in amino acids can lead to structural changes, right? That could make something more fit to survive or less fit to survive. Knowing that this in turn can lead to evolution, the changing of a species, the changing of a creature from one form to the next as lineages as individuals inherit genes that have been mutated. We can take a look at a species, now remember that in a species, another word for species, a more general term for species or a group would be taxa, right? So species is a specific taxa. We could do genus, right? Or we could do order, but no matter what, it's a taxa. So there's a chimpanzee, there's a dog, there's a fruit fly, and there's a roundworm. Now they're looking specifically at an enzyme. Now, can I ask you, just looking at these four, quickly, which do you think are most closely related? Why would you say fruit fly and roundworm, the two most closely related? Huh? A roundworm is not an insect, right? But it's a good try. It's an invertebrate, right? So they're both invertebrates, but an arthropod, or an insect, has 6 legs, has an exoskeleton. Roundworms don't have exoskeletons, and they have no skeleton at all, actually. And they have, they have no limbs, not 6. They don't have 6. They have none. Right? It's a worm. Does that make sense? All right. So, but they are both invertebrates. So they are more closely related than they are to either one of these. These two are very closely related.
Oh, because jaws.
They have jaws, but more importantly, they have vertebra. They have backbones. We call them vertebrates, right? Fish are vertebrates, dogs are vertebrates, chimps are vertebrates, right? Yeah, humans are vertebrates. Yeah.
Do they both have placentas?
Yes, both have placentas. So they're both placental mammals. On top of that, on top of being vertebrates, they're both mammals. So we would expect that these two are the two closest. That's my expectation. Does that make sense? If you're going to pick two that are closest, are these two very close? I'm not. Yeah, but I guess they're closer than they are to chimps or dogs. But are they close to one another? I don't know. Maybe they're equally distant from one another evolutionarily, many generations separated. When I say distant, I mean in time, right? Because things take time to evolve. So how many generations did it take to get a fruit fly versus a roundworm? Or a chimpanzee versus a fruit fly? I don't know. But the common ancestor between these two are probably a lot closer than the common ancestor between these two. Does that make sense? If I look at where on that line, if I'm looking at this line that I'm going to end up with, right? If I have this line, if I have a branch, I don't know, doing glycolysis at all maybe is here, glycolysis period. Having glycolysis 3-phosphate dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase. Maybe having that at all is here, and maybe these guys branch off and you get a chimp and a dog. But maybe all the way down here you had the roundworm, and over here you had the fruit fly. Where these two share a very close common ancestor, these don't really share a common ancestor very closely. That's one possible drawing. We don't have any evidence for that yet. But do you see what I'm saying? Is it making sense to you? Because you need to understand the concept that two things can have a close relationship, a close common ancestor, closer. When I say close, how long ago did chimpanzees and dogs have a common ancestor? How long ago do you think they had a common ancestor? Yeah, probably a million, into the millions, right? I don't know. I don't know the number, but I would say, yeah, probably millions of years ago, right? Some, not too many millions, because too many millions of years ago, too many millions of years ago would have had a common ancestor, but when's the closest common ancestor, right? All right, so I'm going to take a look at the DNA similarity and gene percentage. Gene percentage. What percentage of that gene, specifically what gene are we talking about? The gene for what? What gene are we talking about in this problem set? Humans? Humans, first of all, humans are not a gene, and then secondly, humans are not mentioned in here at all. What gene are we talking about? Gene percentage similarity. That's right. The similarity between of the gene for glyceraldehyde 3-phosphate dehydrogenase. There's this enzyme, G3P. There's G3P dehydrogenase. How similar is the gene, the DNA, that makes obviously the RNA that finally gets translated into the protein that's the enzyme? How similar is this DNA among these species? What percentage of the DNA is the same? The same to what? To what? Read the problem. Did you read the problem? Barron already said it. There's a comparison to humans, right? How close are these to humans? Yes? So if we have 99% similarity to humans in chimps, 99.6%. All right. The dog has 91.3%. Remembering that humans are vertebrates, dogs are vertebrates, fruit flies and roundworms are not, that kind of makes sense, doesn't it? we would expect that we're closely related to dogs and chimps since we're all mammals, and that the percentage, so we'd expect that the percentage of DNA would be similar. Fruit fly is 72%, very different, 72.4% for that gene. And a roundworm is 68.2%. So roundworm's even further out than a fruit fly as far as similarity to humans. And protein similarity, if we look at the protein, now that's the gene, and remembering that the gene is the sequence of DNA. Remember that that DNA may not code for difference, right? We have silent mutations. So the DNA may be different, and you might still end up with the same protein, right? Do you remember that? Do you remember that the codon redundancy, that we can have two codons that are different, two RNA codons that are different, but they give you the same amino acid? All right? So you can have a protein that is actually the same, 100% the same, even though the DNA is different. And of course here with the dog, it's 95.2%, and here the 76.7%. So you see the DNA is more different than the actual protein. Again, that makes sense if you think about it. All right. So now the question is, how are we going to use this? How are we going to use this to build a cladogram? We're going to include humans. So we have a cladogram. That means you have a line. Here's humans. Where would you put everybody? Roundworm will be over here. There it is. It's just that simple. Roundworm, then fruit fly, then dog, then chimp. So when we look at this, humans and roundworms had a common ancestor back in the day. Probably a lot further back than the common ancestor between chimp and humans. Does that make sense? And these percentages demonstrate that. That seems simple enough, does it not? Yeah.
We don't have to put like those extra dots and stuff with the
In this case, I don't see why you want to. I mean, you could, if you had to. Oh, come on. You could put a line here and say, I don't know. What would you put?
Gene similarity percentage.
Gene similarity percentage. So you might put something like, is it 70%? So this is a little, roundworms are a little less than 70, right? And fruit flies, maybe you put 75%. And dog, maybe you put like 92%. And humans, what, 99.7%. So that anything that's on this side is 70%. Or more. Anything on this side is less than 70%. I don't know. I'm not sure why you would do that. Just to say that this is enough, having the order of how close, looking at the common ancestors, the order, the relationship, demonstrating the relationship between the organisms is enough. Okay? All right, let's look at number 2 here. It says, use the following data to construct a cladogram of the major plant groups. So I'm not going to draw this out. So there's some major plant groups here. What are the major plant groups that they list? Mosses, remember those? Which do you think is going to be the most different of all these? What do you think is, without looking at the numbers, don't look at the numbers. You have mosses, you have pine trees, you have flowering plants, you have ferns. What do you think is the most different? Yeah. Pine trees. Why? That's fair.
Smaller.
That's a fair thing to say. Pine trees are big. They have pine needles, right? They don't flower really, though they do produce pine cones, right? But mosses actually are very, very different. They don't need. They are. Remember what are mosses? What are the things you have to remember about mosses when it comes to your end of course exam? And what's so special about them?
Isn't that like a bacteria?
Say that again.
Is that like a bacteria or something like that?
No, it's a plant. It's a plant without roots. It's a plant without roots. Why would that be important? Do you remember primary and secondary succession? The word pioneer species. They're the first ones to show up. Mosses are the first organisms to show up. Why? Because they don't need soil. All the rest of these need soil. Mosses can grow on top of rocks. Mosses can grow hanging from trees. They don't have roots, so they don't need soil. They take their water right from the air. So mosses are substantially different, and the data supports it, doesn't it? Mosses don't have vascular tissue at all. They don't circulate xylem and phloem. All right, so we're going to have a line. Obviously, it's a cladogram. There's 3 traits. What are they? Vascular tissue, flowers, or seeds. Which is the outgroup? Which group has none of the traits? Mosses. So that tells you how different they are. So the first trait is, let's just go ahead and put which one is the one that has, which trait is do most creatures have? So vascular tissue, so let's go ahead and put vascular tissue, I'm just going to put VT. And I'll put mosses down here. So mosses are the outgroup. They have none of those. They are the one group that's the most, it's the oldest for sure of the plants here. So we have a total of 3 pine trees, flowering plants, or ferns. What is the next trait that the next largest number of organisms have? Seeds. And how many organisms have seeds? 2. So moss is already taken care of, so we don't have to worry about that. But somewhere along this line, we're going to have seeds. But before seeds, what creature has to come off? The ferns have to come off, because ferns don't have seeds. Do you see how we draw it? You ask yourself, which one of these has the next lowest one? Which character, obviously flowers, right? Which creature has seeds but does not have flowers? Oops. So we have pine trees coming off. I'm just going to put PT, pine trees. And which creature has vascular tissue, seeds, flowers, and flowers? Flowering plants. So now looking at these character traits, when we look at the fossil record, if you're a paleontologist, you're a paleobotanist, you're looking for evidence of evolutionary development, and you're digging up rocks in Africa or South America, or up in Canada somewhere, wherever you're digging up rocks, and you're looking for fossils. Which do you expect to see in the deepest part of the rock structure? Mosses. You'd expect to see mosses at the deepest level. Next, you'd expect to see what? If you found some dinosaurs, what would you expect to see next to the dinosaurs, or creatures that came before dinosaurs? what plants do you expect to see? Ferns next, the next deepest level. And then, would you expect to see in the fossil record pine trees show up, and right next to pine trees would be flowering plants? Probably not. Probably you'd see pine trees first, and then sometime later, you'd see flowering plants. So actually, flowering plants are new to planet Earth, relatively speaking. Ferns have been around a long time. Whenever you see dinosaur movies, you always see ferns hanging around. They've been around for a long time. Moss is obviously even longer. All right, so that makes sense, yes? And by the way, the fossil record does support this. Yeah?
So there's this one thing on the fossil record that when you a whole abundance of a single species, and it can also help with human time, like the little creatures that were like those spiral things. I forgot the name of them. But
Nautilus?
probably, but they were like spirals, and they were down, and then they were here before the dinosaurs, and they helped determine in the fossil record. Would humans also be a good marker because there's an abundance of humans?
I see what you're saying. I think I understand your question. If I understand your question is, when you're looking at the fossil record, whenever you see abundance of animals or plants, that's a good marker of an era. And that you're able to say, well, we find all these ferns, and this happened about, I don't know, I'm just going to make up a number, 50 million years ago. So that every time we find in the rock a bunch of ferns, we're going to say it's about 50 million years ago. Is that what you're saying? Yeah, that is a good indicator. The thing is that we have much more accurate measurements called by radioactive decay methods. So we can look at half-life and of the rock itself. So we can see how much carbon is left from back then, and based on how much carbon is left from back then, because it decays over time, right? The radioactive isotope of carbon, I believe carbon-14. Yeah. So in any case, the point is that as if I look, we see how much of that radioactive carbon is left. It can tell us much more accurately how old that rock structure is, and so we've been able to date things that way. But there is further evidence by seeing ferns, a bunch of ferns in that. And yes, are humans a good indicator of an era or of a time? Yes, for sure. If you see humans, Homo sapien, you have a certain time range because we don't see them before. Does that make sense? So we know that humans lasted. If we ever find a human that's not because there's been turnover, right? Not because the land is flipped over because of an earthquake. But if we ever find humans sitting next to a dinosaur fossil, then we got to change our theory. But we have not found it. Nowhere on Earth, in all the digging we've done, have we found a human and dinosaur skull next to each other. And you'd figure that some dinosaur ate some human, right? If they lived together, don't you think? We do find humans and saber-toothed tigers together. We do find woolly mammoth bones next to humans, right? We find fossils of humans, but we don't ever find them next to dinosaurs. That's more evidence, right? It's that stratification that humans came after. We evolved after these other organisms. So, in any case, I'm going to let you do the next 3, 4, 5, and 6 in the last 10 minutes of class. I expect all the rest of them done and submitted by, what would you say? Tomorrow are we meeting tomorrow?
Yeah, we don't have school Thursday.
I know, but are we meeting? Because I know that tomorrow we're not. Some of us aren't meeting. Some of my biology classes aren't meeting. I know today my 6th and 7th period are not meeting because they're doing their pro core. I think tomorrow. Don't you have history pro core tomorrow?
Yeah. Oh,
Is it first period? I think it's first period, first and second. Yeah, I think first and second tomorrow you have history pro core. So I expect this completed, right, and uploaded, and I will upload for you a complete answer set on Thursday. or Friday. Maybe it might have to be Saturday because I need you to remember, I need to print out your spring packets. You need to get your end of course review packets before you leave today or tomorrow. Because I will not be here Thursday or Friday. Is that clear? I will be in Chicago on a NIMSY grant, and so I will need to be absent on Friday, so I will not be here on Friday. I know you have your in the afternoon you have your pep rally, so I hope you have fun. If you need somewhere to be on Friday, I'm not sure. Usually I have because I don't like all that noise, I do have my room open on Fridays for people during pep rally, but I won't be here this Friday, so if you want a quiet place to be, you're gonna have to find another teacher that's staying behind. Yeah?
So, can you send us a copy, like, just
Yeah, I will send you a copy as well, but I'll have them printed. I think you probably want it printed.
Will they be printed by advisor?
Yeah, I'm gonna print them actually now while you guys are working on this. I haven't done, I just want you to print them, okay? And Raven's gonna go get them, so hopefully you'll have them before you leave right now. But Raven's kind of slow this morning, so maybe. I'm just kidding. Slow moving. Do you see how slow she's moving? Look at her.
Oh, I forgot to turn my phone on.
And there it is. Evidence of what I just stated.